For 3 decades, the causal architecture of Alzheimer's disease has been organized around a linear progression. Amyloid-beta aggregates upstream as the trigger, while the hyperphosphorylation of tau follows downstream as the primary driver of cognitive decline. But this model contains a persistent spatial and temporal paradox. Amyloid pathology correlates only weakly with the timing of decline, while the transsynaptic march of tau correlates strongly. There is a physical gap between where amyloid deposits and where tau begins its spread. The specific molecular relay that carries the toxic signal across this vacancy has remained unidentified. Without a confirmed bridge, the transition from one proteinopathy to the other remains a mystery rather than a mechanism. This failure to identify the bridge explains why removing amyloid late in the disease fails to stop the clinical progression. By the time the symptoms appear, the bridge has already been crossed. To find that bridge, we have to look at the microglial cells surveilling the extracellular space. These are the resident myeloid cells of the brain. They are constantly monitoring the surrounding neural environment. In the traditional narrative of neurodegeneration, these cells were treated as reactive scenery, a secondary consequence of damage rather than an active participant in its creation. Recent evidence suggests the microglion carries a specific organelle, the NLRP3 Inflammasome, that actively reads amyloid as a danger signal and translates that reading into the acceleration of tau. This immune sensor is the machinery that converts a slow, localized proteinopathy into a self-sustaining disease that spreads across the tissue. The NLRP3 inflammasome is governed by a strict molecular logic. It requires 2 sequential signals, priming and activation, before it can fire. The first signal, priming, occurs when soluble amyloid-beta binds to the CD36 and TLR4 receptor complex on the microglial surface. This triggers a transcriptional shift in the nucleus, raising the levels of the NLRP3 sensor and the precursor cytokine, pro-IL-1-beta. The second signal, activation, is triggered when the microglion phagocytoses a larger, fibrillar form of amyloid-beta, drawing it into an internal lysosome. The rigid amyloid fibrils rupture the lysosomal membrane, which releases cathepsin B into the cytosol and triggers a sharp drop in intracellular potassium. Through this process, the microglion participates in manufacturing its own trigger. It assembles the amyloid peptide into the exact particulate danger signal required to fire the sensor. Once activated, NLRP3 proteins oligomerize and nucleate the adapter protein ASC. These proteins link together to form a micron-scale filamentous aggregate. This ASC scaffold then recruits and activates the catalytic engine of the system, the enzyme caspase-1. Active caspase-1 creates a bifurcation in the microglion's output. First, it matures pro-IL-1-beta into active secreted cytokines that drive local inflammation. Second, it cleaves a protein called gastromin D. The resulting fragments rush to the cell's surface, inserting themselves into the membrane to form physical pores. This bifurcation provides the inflammasome with 2 different effectors, a soluble chemical signal through cytokines and a physical lesion through membrane pores. When the gastromin D pores reach a critical density, the microglion undergoes pyroptosis. The cell swells and ruptures, violently spilling its contents into the extracellular space. Among the debris left by the dying cell is the ASC speck, the massive insoluble protein aggregate that served as the inflammasome's scaffold. This extracellular speck does not dissipate. It persists in the brain tissue, acting as a prion-like nucleating seed. Soluble amyloid-beta in the surrounding environment binds directly to the ASC speck. The speck cross-seeds the aggregation of the peptide, accelerating the growth of new amyloid plaques. Pyroptosis effectively weaponizes the internal sensor. It transforms a cellular alarm into a persistent extracellular template for further proteinopathy. The ASC speck fills the long-standing vacancy in the amyloid cascade. Its presence acts on nearby neurons, shifting the internal balance of kinases and phosphatases. Specifically, the speck-induced signal suppresses the phosphatase PP2A, which directly drives the hyperphosphorylation and aggregation of tau inside the neuron. Meanwhile, the release of IL-1-beta cytokines target neighboring healthy microglia, spreading the activation signal through the tissue. This cytokine bombardment impairs the phagocytic capacity of those neighboring cells, specifically destroying their ability to clear ambient amyloid-beta. This creates a feed-forward spiral. The inflammasome halts the clearance of amyloid while accelerating the production of tau, manufacturing the exact danger signals it was evolved to detect. It is important to clarify that the inflammasome is an amplifier, not an initiator. It responds to the generic signatures of injury that appear after the first amyloid aggregates have already formed. In preclinical stages, the gain of this inflammatory loop remains low. Microglia clear the amyloid trigger before the sensor fires. But as ASC specks accumulate and tau enters as a second trigger, the system reaches a tipping point. The spiral becomes self-sustaining. Past this threshold, the disease no longer requires the original amyloid stimulus. It propagates independently, driven by its own inflammatory exhaust. This explains the failure of broad anti-inflammatory trials. Those drugs were administered too late, after the tipping point, and they suppressed the entire immune system, including the protective phagocytosis needed to clear plaques. A viable therapeutic requires highly selective inhibition. Using small molecules like MCC950 targets only the NLRP3 sensor or the gasdermin depor, avoiding total suppression. This selective approach closes the pyroptotic amplifier and stops cytokine release, while fundamentally restoring the microglia's ability to clear amyloid. The success of this strategy depends on timing. Selective inhibition must occur in the early prodromal window, before the amyloid-tau relay becomes autonomous. Stopping the neuroinflammatory spread of Alzheimer's requires the precise silencing of the amplifier, while the loop still depends on its initial trigger to run.